Hi everyone, I'm Safiye, Data Science Manager at Recursion.
Hi, I'm JC, Principal Computational Scientist at Genentech.
Today, we'll tell you about our work, biological cartography. Here's our disclaimers. The views we are about to express are our own and may include forward-looking statements. The continued scaling of genetic perturbation technologies combined with high-dimensional assays, such as microscopy and RNA sequencing, has enabled genome-scale reverse genetics experiments that go beyond single endpoint measurements. Datasets emerging from these experiments can be combined to construct maps of biology in which perturbation readouts are placed in unified, relatable embedding spaces to capture known biological relationships and discover new ones. Construction of maps involves many technical choices in both experimental and computational protocols, motivating the design of benchmark procedures by which to evaluate map quality in a systematic, unbiased manner. In this work, starting with biological perturbations, we first proposed the IFAR framework for map building. We then proposed two key classes of benchmarks to assess the quality of a map. We described univariate benchmarks evaluating the quality of individual perturbations and multivariate benchmarks evaluating how well the map can recover known biological relationships from large-scale public data sources. We demonstrate the application and interpretation of these benchmarks through example maps of single-cell RNA-seq and imaging data. Before we go into the details of our work, here is the team who worked hard to make it happen. This was a great collaboration between Recursion and Genentech researchers aiming to guide the field in building and benchmarking maps of biology by introducing a framework which can be used in such efforts by others. Let's now look at the IFAR framework more closely. The first step is embedding perturbation units. We call the smallest experimental entity that is measured in a map context a perturbation unit. This can be a single cell or a well with hundreds of cells. The embedding step is aimed at creating a vector representation of the experimental screening results. Intermediate layers of neural networks are commonly used to generate embeddings for unstructured data like cell images. Linear dimensionality reduction methods like principal component analysis are common for structured data such as transcriptomic profiles. However, nonlinear dimensionality reduction techniques based on neural networks have been found to be effective there as well. In any experimental screening process, some perturbation units will not satisfy the predefined quality criteria and need to be filtered out. This filtering can occur before or after embeddings are generated. Examples include wells with too high or too low pixel intensity in a cellular imaging screen, or perturbation units that are not distinguishable from the controls in terms of their readout or embeddings. Then comes the alignment, which aims to correct for batch effects. A batch effect is a systematic effect shared by all observations obtained under similar experimental conditions, such as microscopy acquisition artifacts or incubation times. These potentially confound the interpretation of desired biological signal from the readouts. A simple approach for alignment is to use control units in each batch to center and scale features in that batch. Another linear method aligning not only the first-order statistics, but also the covariance structures is typical variation normalization, or shortly TVN. Nonlinear methods based on nearest neighbor matching or conditional variational autoencoders have been particularly successful for the alignment of single-cell transcriptomic data. There are typically multiple technical or biological replicates representing each perturbation in any type of map data, meaning the same perturbation may be applied to dozens of wells or hundreds of cells. Aggregation of these replicates is critical for a robust final representation of a perturbation. Coordinate-wise mean and median aggregation are commonly used methodologies here. The final step in map building is identifying relationships between biological entities, such as gene-gene interactions arising from protein complexes or signaling pathways. Computing distances between aggregated perturbation representations is commonly used as a proxy for relationships, where smaller distance means a stronger relationship. These distances can also be used to visualize the global structure of perturbations through further dimensionality reduction techniques such as UMAP. We can use different methodologies in every step of the IFAR pipeline, and we can perform the steps in a different order or multiple times. We divide map benchmarks into two types, which we call univariate and multivariate benchmarks. Univariate benchmarks assess the ability of an IFAR pipeline to recover signal on individual perturbations, utilizing the replicates of a perturbation after alignment. Multivariate benchmarks, on the other hand, utilize the relationships between aggregate representations of perturbations to evaluate a map's ability to recover non-biological relationships.
Let's first talk about univariate benchmarks. These assess the reproducibility and robustness of the representations of the individual perturbations in a map. We demonstrate two metrics here. The first one is the consistency of the perturbation profile across replicates as quantified by the average cosine similarity between replicates. The second one is the magnitude of the perturbation effect as quantified by the energy distance. For both metrics, we can assess the significance of the effect by assigning a p-value based on random sets of perturbations as empirical null. The second type of benchmarks, multivariate benchmarks, focus on known relationships among perturbations. Capturing these is important since a typical use case of a map of biology is to identify novel, biologically relevant relationships between pairs of genes or between genes and small molecules. In this work, we focus entirely on pairs of genes. There are two main data sources for these multivariate benchmarks: databases of pairwise relationships and of gene clusters. Pairwise relationships refer to known gene pairs that directly interact in a signaling pathway or a protein complex. Gene clusters refer to sets of genes of any size that are involved in the same biological process, pathway, and again, protein complex. In both cases, as a benchmarking metric, we put the recall at 10%, or more precisely, the percentage of annotated gene pairs that we capture within the most extreme 10% of pairwise relationships, considering 5% from both tails of the pairwise cosine similarity distribution. We apply these benchmarks to two orthogonal datasets: the recently published genome-wide perturb-seq data by Replogle et al. And the recursion phenomic data. For each of the maps, we're considering two pipeline choices. On the left, we show those for the transcriptomic map. The first option is a slight variation on the pipeline used in the Replogle et al. Paper. We call it the PCA pipeline, and it uses linear dimensionality reduction and batch correction techniques. The second option is called SCVI pipeline, and it uses a conditional variational autoencoder that simultaneously provides nonlinear dimensionality reduction and batch correction.
For the recursion phenomic data, we compare two pipelines with different alignment strategies. The center scale pipeline centers embeddings per batch at 0 and scales to unit variance based on the controls in the batch. The second one, titled TVN pipeline here, uses typical variation normalization, which decreases the impact of the dimensions that capture variance among the controls.
Depending on the choices made in the IFAR pipeline, you can get different levels of recapitulation of known biology. Here are our univariate and multivariate benchmark results for the 4 maps discussed on the previous slide: 2 transcriptomic maps and 2 phenomic maps. We report our results over all genes and skip the filtering step of perturbations without a significant effect. This is to have an apples-to-apples comparison using the same set of genes between the pipelines. For pairwise relationships, we consider 2 publicly available sources: reactome protein-protein interactions from complexes with at most 4 proteins, and signor signaling pathway interactions. For cluster identification metrics, we consider 3 publicly available sources: reactome gene sets from the MSigDB C2 collection, signor pathways, and quorum protein complexes. Results on the transcriptomic data for different embedding and alignment choices are shown on the left-hand side table, showing a slight advantage of using the SCVI pipeline compared to the PCA one.
For the recursion phenomic data, we report recall results relative to a convolutional neural network trained to be partially resilient to batch effects. We see that an alignment step by TVN or center scale leads to a considerable increase in a majority of the metrics compared to the baseline, and TVN has a clear advantage over center scale.
On the previous slide, we looked at the results globally across thousands of pathways. To provide a more granular perspective, let's now look at how the two transcriptomic maps compare in terms of the recall value per cluster on the quorum dataset, as shown in this scatter plot. Consistent with the summary metrics, the SCVI pipeline performs better for most of the clusters, as represented by more of the points falling above the diagonal than below. For some selected complexes, we show the cosine similarities between pairs of genes in a split heat map on the right. Above the diagonal are the relationships using the PCA pipeline, and below the diagonal are the relationships using the SCVI pipeline. While most relationships look very similar, the BRG1 associated complex in dark green exhibits a clearer clustering structure using the SCVI pipeline, as suggested by the recall metrics for that complex.
As another example of the known and novel biology identified by the benchmarked EFR pipelines, we examined the cosine similarity structure for the integrator complex, which was also explored in the genome-wide perturb-seq paper. Here we look at two split heat maps, one for the transcriptomic maps and one for the phenomic maps we built. On the left heat map, above the diagonal are relationships from the PCA pipeline, and below the diagonal are the ones from the SCVI pipeline. And on the right heat map, above the diagonal are relationships from the center scale pipeline, and below the diagonal are the ones from the TVN pipeline. Non-biological structure of the integrator complex is presented on the right. We see that both data types and all four maps accurately identify the modular structure of the integrator complex, and they all identify a previously unknown gene, C7orf26, as part of this complex. This gene was officially renamed INS15 earlier this year, after two papers, including the genome-wide perturb-seq paper, proposed this gene as part of the integrator complex. Let's end with a summary of our contributions here. In this work, we describe a framework for systematically constructing whole genome maps of biology and benchmarking their performance globally. As a demonstration, we present several map options built using two orthogonal data types: single-cell transcriptomic data where perturbations are based on CRISPR interference, and array-based phenotypic screening with CRISPR knockout. Results demonstrate the impact of different processing pipelines and metric choices. This framework can be used for any large-scale biological map building and benchmarking effort, regardless of data types. It can be expanded to include settings where additional perturbation types or assay variables are assessed. We hope that this framework will be generally useful as these large-scale map building efforts become more common. And note that both Recursion and Genentech are hiring. Take a look at our careers pages.